Hello, everybody. My name is Zaira. I am a PhD student of the Colegio de Postgraduados in Mexico. And well, I am glad to share my research with all of you. So let me use my pointer. Okay, I start emphasizing this phrase because in some forest types, the organic matter accumulates in one or more thick organic ridge at horizon at the surface of the soil. And this forest floor is important to energy budgets and nutrient cycling. And actually, previous estimates have determined that that the forest floor can store up to 5% of the global forest carbon. And even though the size of this pool may seem minor compared to mineral soil or vegetation, the forest floor is an essential component in plant-soil relationships because it participates in a lot of ecological or biogeochemical ecosystem processes, and it is more sensitive to changes compared to the mineral soil. The amount of nutrients stored in the forest floor doesn't remain fixed through time and space, and this variation is especially important for managed ecosystems. Forest management, especially harvesting and thinning, can change the original composition and structure of a forest and shift carbon into the tidal pools. And for sure, any forestry actions will affect the nutrient balance of forest floor and will have implications for the long-term nutrient storage capacity. This can happen through different mechanisms, including altering the quantity and quality of the trial carbon inputs, changing the composition of the soil microbial communities, modifying the soil conditions that drive microbial processes, or by fragmenting and mixing the organic matter with the mineral soil. And clearly, these changes mediated by the forest floor will directly affect decay rates and carbon fluxes. So certainly, all the interactions I mentioned before make it difficult to monitoring nutrient storage in the forest floor in managed ecosystems, but including the effects of management practices in carbon and nitrogen estimates with a rigorous, conclusive, and statistically valid approach is difficult because of sampling and measurement challenges and the inherent spatial variability in forest floor properties and the large number of environmental variables required to detect the variation across the landscape or over time. And one approach is modeling the relationships with environmental factors associated with the status of vegetation and landscape conditions, like tree species, canopy structure, topography, or climate. So far, many studies aren't based only on measured soil properties or in the use of linear models, but also they've used several statistical and spatial approaches that are able to modeling complex nonlinear relationships or interactions, which is more important. However, only few studies have applied these techniques in forest floor assessments, and this knowledge will assist regional or national efforts to estimate nutrient storage or in forest soils, identify areas with potential fire risk, guide long-term management activities, especially for carbon monitoring and reporting. So one of the most recent techniques of digital soil mapping is the random forest algorithm. This machine learning technique is recognized for two advantages. First, the model can deal with multicollinearity or non-normal distribution of their variables. And the second one is that random forest can define non-linear relationships between external factors and dependent variables. So in this study carried out in a temperate managed forest, I estimated carbon and nitrogen stocks of the forest floor. The spatiotemporal patterns were modeled as well as their relationship with external variables using random forest. This information will be useful to identify the parameters affecting carbon storage and adopting appropriate long-term management practices. In addition, the obtained maps can also be used as an input for mechanistic simulation models for nutrient cycling and climate change. The research was carried out in the intensive carbon monitoring site located in the state of Hidalgo in central Mexico. The land of the study site belongs to rural forest communities, and since 1980, the owners implemented a timber management program following the principles of the silvicultural development method. And their forestry actions have created now a 40 years chronosequence of even age stands of Pinus patula through the seed tree regeneration method. Thinning is the most common intermediate silvicultural treatment. The sampling was carried out in 2013 and 2018, and all measurements were made in 40 permanent sampling plots, as you can see in the 3x3 polygon. The SMICA Topisco follow a sampling scheme and developed for similar monitoring sites in the US, and all sampling protocols were consistent with the design of the Mexican National Forest and Soil Inventory. Then I decided to split and assess the forest floor into 2 layers, the fresh litter material and the fermentation layer. And in laboratory, I estimated the carbon and nitrogen concentrations and stocks, as well as their respective stoichiometric ratio. The models were built as most parsimonious as possible. So after an exhaustive review of predictor variables, I only chose 10 of them, representing the topographic and canopy structure conditions. The information was obtained by different sources, such as LiDAR metrics and forest inventories. And some of this information comes from previous studies. So 2 random forest models were created, one for 2013, and the procedure was repeated for the 2018 dataset in order to obtain the estimates by forest floor layer and evaluate its performance in the forecast process. We create a new dummy predictor variable, which represented the decomposition stage of the forest floor with 2 levels. And finally, the models were validated by K4 cross validation, and this was our model outputs. Here, I show you the average values of each evaluated nutrient. As we can see, there are remarkable differences both between forest floor layers and years. And the highest stocks were recorded in the most decomposed layers of both years. And but in 2013, the nutrient storage was greater than in 2018. And well, regarding the stoichiometric ratio, regarding the stoichiometric ratio, the highest values in 2013 suggest a slower decay of organic material than in 2018, and which could be a reason for a higher accumulation of dead good organic matter in 2013. The figure here show the spatial predictions obtained with the random forest algorithm. Here, I emphasize that both carbon or nitrogen stocks exhibited remarkable differences in spatiotemporal patterns and highlighted their dependence on biophysical variables and forest disturbances. The topographic and canopy structure conditions were selected as the principal predictors, since the variability in climatic conditions within the study area is quite small. The spatial patterns were mainly explained by basal area, stand age, and tree cover, which in turn are highly controlled by forest management practices. And and the regions with the highest nutrient stocks on the forest floor, represented by blue colors, are found in the stands that have been recently harvested or in those with high tree cover values. So the areas with a higher stoichiometric ratio represented by dark green colors are possibly nitrogen limited, and therefore it have slower decay rates and accumulate more organic matter. On the other hand, when we evaluated the model performance, the models of stoichiometric ratio had the less uncertainty compared to the maps of carbon or nitrogen. However, all models explain more than 60% of the variance, which is a high value. And despite of the difficulty to extrapolate the measures made from a plot to landscape scale, the model performance was superior to other studies that have used more predictor variables and higher sample densities. Here we can see how carbon and nitrogen stocks change as a function of stand age or basal area. We found that in the first 5 years after harvesting, nutrient stocks tends to decrease, maybe by an accelerated decay. And then as the stand reaches maturity and the canopy closures, the carbon stocks tends to increase again, maybe by the increasing of leader inputs. And this trend was similar for nitrogen stocks. We also observed that reductions in basal area have a drastic impact on the amount of nutrient stored, especially for nitrogen. Both curves show the impact of removing vegetation and prove that forest floor layers are strongly sensitive to changes in the canopy structure. Well, regarding topographic variables, the carbon and nitrogen stocks tend to be higher as the slope increases, and maybe this interaction is not strictly direct and instead reflects the effect of other variables that weren't included, such as rainfall or wind. Well, there are some implications in the modeling approach we use. First, the assessment of nutrient stocks by forest floor layers had a strong impact on model performance. And one reason was that both layers exhibited an heteroscedastic statistical behavior and a special autocorrelation. As a result, we noticed a limitation in the model performance, especially an underestimation, which has also been reported in studies at regional or national scales. Some further analysis suggests that this limitation is due to the differences of residual variance, and random forest cannot fully represent it. However, we are trying to see if this model disadvantage can be solved through additional residual models or using other geostatistical tools. Finally, the advantage of random forest is that it can reduce the sampling effort because it works well with multi-source data as long as the variable selection are appropriate. And obviously, this is linked to the parsimony principle. Well, evidently, these results highlight the importance of using multiple methods for integral estimates of forest floor properties through time and space. And our results can help to promote effective forest management strategies for protecting areas with high levels of organic matter, or maybe for preventing wildfires or soil erosion. Well, that's it. Thank you so much for your attention. Any questions?
I'm a NSF EPSCoR postdoc at the Oklahoma State University, and today I'll be presenting on shifts in fine root dynamics along a hill slope in a central Pennsylvania forest. So here's a picture from my site, and these flags going up sort of indicate the gentle slope here, but the slopes get a little bit more steep in other areas throughout the catchment. But what I'm trying to illustrate is we're standing here in the valley floor. So as you go up in this picture, eventually you would get to the ridgetop, which is somewhere up there. And a lot of root research has been done primarily in flat areas. So what is the effect of topography on fine roots, especially in forests? So we know from previous work out of Shale Hills, which is the area where this study was conducted, that from this group, that standing where we were in that picture, virtually, in the valley floor, as you go up slope to the ridgetop, the soil moisture over time comparably is lower. So you have higher soil moisture in the valley floor. And other groups out of Shale Hills have shown above ground effects. So this group, Smith et al. 2017, and this is Shale Hills again, showed that above ground carbon storage in the woody increment increases going from cold colors to hot colors. So blue is relatively low, red is pretty high. Tends to be higher in the south facing aspect, as well as in these cutout regions going upslope, which are swales. So areas of, like if you were walking in the catchment, that would be kind of a divot, so where leaves and water tend to collect. And this figure, I think, from their paper also helps demonstrate this a little bit more. So in the swale side, the dots get bigger with more carbon storage in the wood. And then I have the species listed there for those of you that are interested in the acronyms. So this led us to the question, do these above ground trends continue below ground, particularly for fine roots? So I'm going to walk through the fine root dynamics that we looked at. So we looked at fine root production, which is when a root is produced, fine root standing crop, the number of roots in a given area, fine root turnover, which is the number of times a population of roots is replaced over a period of time, usually a year, and then fine root mortality, which is the number of times a root dies. Well, the number of roots that die, really. So for each of these dynamics, we expect that with increasing soil moisture for a given topographic region going from dry regions to wetter regions, we expect a quadratic trend or a nonlinear trend. So increasing soil moisture is beneficial to these processes up to until a point where the soils typically begin to get anoxic and detrimental to roots dynamics overall. And I forgot to mention at the beginning of the talk, this is all published in Plant and Soil if you're interested under the same title. So feel free to look it up later if you want. To give you an idea of how we sampled within the site, this is a figure from one of my previous papers where we looked at fine root lifespan, and that's, I'm going to talk about that manuscript at this time. So elevation is lightest at the lowest elevation and gets darker as you go up to the ridge top. We had 50 macroplot sites where we had mini rhizotron tubes installed, as well as at these teal diamond sites, soil moisture sensors. The blue sites represent valley floor sites, green are the swale sites, red, mid-slope planar, and then white is the ridgetop sites. So I mentioned we use soil moisture sensors. So we use TDR sensors that we installed at 20, 40, and 60 centimeters horizontally. They measure soil moisture by basically the 3 metal prongs, and they measure the amount of soil moisture in between the prongs. Within all of those dots that I showed previously, there were 5 mini rhizotron tubes. These were tubes installed on the ground in 2014. We sampled 3 tubes on a monthly basis to full depth, and 2 of those tubes on a biweekly basis down to 45 centimeters. So that's only the first half of the mini rhizotron system for those of you that are unfamiliar with it. The second half, this is the system that we use, which is a laptop with an ATV battery under it, connected to what is essentially a camera on a stick that has a lighted source. And so we can get a continuous soil profile with these pictures. And what these pictures look like is this. And so this is what the scale of the mini rhizotron image is. And this is a nickel for those familiar with United States coinage. But for a more tangible image, this is one of the hundreds of thousands of images we collected. So this is what would be a first order root, which was the roots that we tracked. So no other roots branching off of it. We used Rootfly software through Clemson. So we had to manually track these. So we would, on software, place a dot here and place a dot here, and the software would give us the root length. We would estimate root diameter through the system, and so on and so forth. With that, I will get into our results. So fine root production and mortality, we did not find any significant results. However, for standing crop, so average standing root tips or standing crop, number of roots, natural log transform for the different topographic regions, ridgetop, mid-planar, swale, valley floor, so dry to wet, with increasing year being the darkest bar to lightest bar, so 2016, 2017, 2018. we found that in the mid-slope planar regions relative to the valley floor regions, there was less of the standing absorptive roots or fine roots. When we looked at fine root length standing, also natural log transformed, we found that both swale and the valley floor region were significantly higher in terms of root standing length relative to the mid-slope planar region at the 95% level. When we looked at the marginal level or the 90% level, valley floor was also significantly higher than ridge top. This figure, we're looking at contrast. So we've got the wetter regions on the left, the drier regions on the right, regions that are insignificantly different from each other cross the 0 line. And we're looking at root tip turnover. So what I'm trying to show here is that the drier regions, the ridge top and the mid-slope planar, typically have higher root tip turnover. And this just represents marginally 90% than the wetter regions. And so now I wanted to look at the year effect. So similar to that previous figure, we've got wet years on the left, drier years on the right. Production are the square lines, mortality are the lines with the dot in the center, standing crop are the lines with the filled-in triangle, and then we've got the diamonds at the top, which are turnover. And so what we saw for root tips is that production, mortality, and standing crop tended to be higher in the wetter years. Surprisingly, we found that root tip turnover, on the other hand, tended to be higher in drier years. Additionally, we found different trends in root length. So with the exclusion of root length turnover, which was similar to root tip, root standing crop was insignificantly different whether it was wet or dry year, but there was more root length mortality as well as root length production in the drier years. And this leads me to the discussion. So at the top of the talk, I said that there were strong topographic signals above ground, but we did not find that they continued as strongly as previous studies have shown below ground. And we also found no evidence of a quadratic relationship based on topographic position and the soil water structure that goes with it. Additionally, I didn't really cover this much in our methods, but we really did look at a lot of site level variables. And again, feel free to reference the paper to see what we covered. I'm happy to cover some of that now, but none of those were found to be significant. So what we did find was that wet regions of the landscape tended to employ a conservative root strategy. So that is, the roots that are produced tend to be held, and that goes for root length too. And drier regions, on the other hand, tend to avoid bad conditions or harsh conditions for roots. So they produce roots when conditions are good, let them die when conditions are bad. And unlike what we showed topographically or spatially, year showed strong effects on pretty much all the root dynamics. And with that, I have a lot of acknowledgements to make. These groups funded my research, and the NSF EPSCoR allowed me to present today, as well as the Eisenstat Lab and the host of undergrads, lab managers, and visiting scholars that helped me. These are the citations I had at the top, and with that, I will take any questions.
Hi, everyone. My name is Claire McPullen, and I'm a master's student at the University of Victoria. And my co-author for this project is Marty Groenevetter here from the Ministry of Forests. And our research is looking at ectomycorrhizal fungi species that are endemic to the Pacific Northwestern coast and what might be special about them. So the literature shows that if you take a look at any typical regional species pool, you'll find a composition of both generalist and specialist species, with generalists having this wider niche breadth and specialists having a more narrowed one. And the most commonly accepted ecological model shows that both of these groups or strategy types have the same kind of area under this curve when you're looking at fitness versus niche breadth. And you see generalists spanning this larger area of niche, and specialists holding this fitness advantage in this narrow range of what they call suitable conditions. And those can be biotic or abiotic. And of course, this can occur across a degree of ranges. So from a resource specific level to a wider scale of environmental gradients, as well as temporal, like throughout succession. So wherever you can identify these parameters here, we should theoretically see a subset of the species pool that have this greater fitness or hold this higher fitness. So what I'm exploring is how this theory applies to ectomycorrhizal fungi communities. So generalist and specialist strategies are pretty well reported in ectomycorrhizal fungi community composition. Generalists are typically defined as those that demonstrate very little turnover as you're moving along a nutrient gradient. And they also tend to be reported to dominate in these younger seral stands in younger forests. So an example of that would be Cenococcum geophyllum. It's just like ubiquitous fungus that you can find everywhere in a whole range of different soil profiles all over the world and across different levels of forest succession. And a specialist fungi, on the other hand, would be one that demonstrates really high turnover as you move along a nutrient gradient. And these are always found in association with mature stands, so appearing later in succession. And an example of that would be Lactarius substriatus. And this is a phosphorus specialist fungi that has only been found on really low phosphorus soil sites. And an enzyme assay of it looked and saw really high levels of these phosphorus mobilizing enzymes. So if you were a tree living on a very low phosphorus site and you were to recruit fungal partners to help you with your growth and development, you'd want to be choosing Lactarius substriatus over Cenococcum geophyllum, because that Lactarius species, being more adapted to those low phosphorus sites, is better able to accrue that nutrient and is going to provide superior nutrition than this more generalist type like Cenococcum geophyllum. So when we're interested in conserving or assessing the health or quality of ectomycorrhizal fungi on a community scale, being able to identify this group of like higher fitness specialist species is important. And identifying them might not necessarily be captured in something like a diversity index, which is the kind of commonly used method to infer the health of these communities as it pertains to forestry. So we want to identify these specialists because it's important to tree nutrition. So the next logical question is, how do we go about doing that? So some currently existing methods are enzyme assays. So you can look directly at the enzyme activity levels and expression, different species of nutrient acquiring, mobilizing enzymes. You can also use MIFE, so using microelectrodes to look at that fungal root interface and see the ion exchange to see the rate of nutrient transfer. You can also use sporocarp nutrition. So that idea is based on this 2018 study that looked at ectomycorrhizal fungi stoichiometry in relationship to Douglas fir host stoichiometry. And it showed that there is this sort of direct proportional relationship, suggesting that you can use these sporocarps as a direct indicator of what the tree is receiving. But all of this said, when you consider the vast diversity of ectomycorrhizal fungi species, so taking in a single watershed, up to 360 species have been reported, identifying the superior species on this sort of scale, and especially considering how these compositions can change as you move into different regions, this inductive method might be a challenging approach to being able to identify this sort of group of species. So my project tests the efficacy of using this kind of top-down approach to identify these superior fungi and asking if we can use geographic range as a predictor. So we do see a lot of variation in range between ectomycorrhizal fungi species. So you see some that are typically quite locally bound, and then another subset of the population that'll have a range that spans many different environmental ecological zones. So what we were hypothesizing was if perhaps this model can be applied to this group of fungi, and this range of suitable conditions could be outlined or bound by an ecological zone. And what we were most interested in was the temperate rainforest. So the temperate rainforest spans the Pacific Northwestern coastal region from Alaska down to Northern California. And it outlines a very unique biotic and abiotic niche. So very unique soil profile and climate from the surrounding zone. So the climate's much milder and very, very heavy precipitation year-round, about 3,600 millimeters of rain. It's home to unique biota, like species of western hemlock, Sitka spruce, red cedar, all have these distributions that are contained within this coastline here. And this heavy rainfall causes a really unique soil profile. You get this weathering occurring. So we have much more acidic soils with relatively high nitrogen compared to other forest soils. Is in a quite available form, lots of nitrate, and very low available phosphorus. It's all bound up. So because this region is so distinct, we thought that the fungi that might only be found here might possess an adaptive advantage to those that are found in multiple zones. So that is the research question. And we tackled that by looking at some plots on the southern tip of Vancouver Island here in the very wet western hemlock zone in pink. And we looked at 5 different sites using a split plot design of 100% western hemlock and Sitka spruce plots. They're about 50 years old. And at all of those plots, we searched for all of the fruiting bodies and also dug up root tips. And those fruiting bodies went on to be used for a nutritional analysis, which would be to confer the competitive ability of that particular species. And the root tips went on to generate abundance data to characterize the larger community. And to identify the species, we had the ITS regions sequenced. And then to characterize range, so with the growing availability of molecular tools, this online database called UNITE is used to be able to kind of hold all of these fungal species recordings. So you can put the ITS region into this blast box here, and it can identify the species for you. And you can see all of the different places where that species has been recorded. So using this method to characterize the range, if a species popped up that had a range that spanned a larger area like this, I call that a cosmopolitan. And one that was only found in the Pacific Northwest coast was an endemic. And a good chunk of our species were actually located had only ever been found on the southern tip of Vancouver Island, which was interesting. So to analyze this data, we used linear mixed models to test the effect of fungal range, so the endemics versus the cosmopolitans, as well as the host species. So the sporocarp was found on a spruce or hemlock plot, and how that influenced the sporocarp nutrient concentration. Also looked at how the fungal community composition differed between the tree host species, although that's not going to be the focus of this talk, as well as the phylogenetic breakdown of endemic and cosmopolitan fungal species. So in total, for sporocarps, we looked at about 37 different species were found. 18 of those were cosmopolitan and 19 were endemic. It was pretty even along all of the different sites. And we found significantly higher nitrogen, potassium, and magnesium in the endemic community. And you can see that's fairly consistent across all of these different sites here. And the effect size was much stronger for nitrogen, it was 0.24. And the host species did not have any significant effect on the sporocarp nutrition or any of the nutrients. And sporocarp phosphorus did not show a significant relationship with host range. P was 0.07 and a lower effect size. That said, there was a significant relationship between sporocarp nitrogen and phosphorus found, and relating this to a stoichiometric line of a ratio of 15 from nitrogen to phosphorus suggests that optimal ratio. And you can see that a lot of the endemics fell below that line. They had this higher nitrogen to phosphorus ratio that in a previous publication was associated with higher expression of phosphorus acquiring enzymes. And the idea there is that because the soil has such available nitrate, that fungi that are adapted to just being able to uptake that could be preferentially allocating the energy that would have been used for that nitrogen acquisition to acquire the more energy expensive phosphorus, although this is just one interpretation. And our root tube data showed that there wasn't a strong phylogenetic signal for endemism, and that these species were pretty consistently found throughout different families. The Anosabaceae family showed a much higher proportion of endemics, and the Cortinariaceae family was largely cosmopolitan. But in conclusion, the endemic ectomycorrhizal fungi community seem to be better adapted at exploiting nitrogen, magnesium, and potassium, and potentially phosphorus. And the endemics seem to be phylogenetically dispersed and making up a slightly higher proportion of the Anosabaceae family. But all in all, it seems that these species ranges could be a useful kind of quick top-down method to identify these higher quality ectomycorrhizal fungi community members. And my acknowledgements to the Ministry of Forests and the Center for Forest Biology at University of Victoria. Thanks.
I'm Kathy Fahey. I'm a postdoc at Algoma University. And today I'm going to talk about a project we've been working on looking at the role of soil fungi in the diversity productivity relationship under contrasting water availability. So it's been known for over 150 years that diversity may lead to may have positive connections to productivity. And decades of experiments have supported this idea, and only more recently has this been demonstrated experimentally in forests. And diversity can also improve drought resistance in forests. So for example, in this study that showed nearly 60% of studies have seen a positive relationship between diversity and drought resistance. But even after all of this time, it's not really clear what the mechanisms of these positive interactions are. And so first, I wanted to go through what our baseline null hypothesis is and the possible mechanisms that might be occurring. So in this figure, I'm showing monocultures of 3 different plant species, tree species, and their productivity as they're grown in monoculture. And under our null hypothesis that there are no diversity effects, we expect that the productivity of these 3 species grown in a mixture is going to equal that, approximately the average of the productivity of each of the species in monoculture. And so that's our null hypothesis. But what we can also have is complementarity. And so under complementarity, we would see that species produce more than we would expect under those 3 species in monoculture when they're grown in a mixture. And so there are a variety of different reasons that this can occur. And some of them are driven by microbial drivers in the soil. And so the first most commonly cited mechanism is resource partitioning or niche complementarity. And this can be driven by, for example, mycorrhizal communities in the soil, which can allow plants to access different resource pools, such as organic and inorganic nitrogen, leading to resource partitioning and complementarity. We can also have mycorrhizal networks in the soil, which can connect tree species below ground and lead to sharing of resources and thereby leading to abiotic facilitation. And so we can look for these facilitation interactions by assessing the magnitude of complementarity under high and low stress conditions. For example, under high or low water conditions to look at complementarity for drought. And then finally, we can have biotic feedbacks that lead to complementarity. And so biotic feedbacks with, for example, soil pathogen accumulation, we can have species-specific and density-dependent effects of soil pathogens, and these would be reduced if any particular species is at lower density in these polycultures, and therefore the pathogen effects would be lower compared to in monocultures. And so this would lead to complementarity. And so with this background that microbes might be really important for complementarity, we wanted to ask the question, do soil fungi contribute to the diversity productivity relationship in forests? And if they do, what components of the fungal community, for example, mycorrhizae or pathogens, are driving this relationship? And are the relationships with soil fungi and diversity productivity, are they context dependent, for example, based on resource availability? And so in 2013, Bill Parker set up an experiment based on the International Diversity Experiment Network with Trees design, the IDENT design, in Sault Ste. Marie, Ontario. And he set up 6 different common tree species in Northern Ontario that have a range of different functional traits. And then there are 22 different combinations of 1, 2, 4, and 6 species. And these were replicated in 8 blocks. And then our experimental design separated these 8 blocks into high and low water availability using either gutters, as you can see in the picture here, or irrigation during the growing season. And then what I did is I took soil samples in 2020 and sequenced the ITS region and to assess the fungal community composition in each of these different plots. And then the results that I'm going to show are based on structural equation models, looking at the variables of water availability, so that's that drought or irrigation treatment, functional identity of the tree species, and species richness. So functional identity here is basically just looking at the average trait values of each community. So this includes traits such as leaf lifespan and photosynthetic rate. And then the fungal variables that I looked at were fungal community composition. So there's 2 of these here, fungal community 1 and fungal community 2, because these are based on the 2-axis of an NMDS ordination. So it's basically just representing the whole fungal community. And then we also included pathogen richness and ectomycorrhizal richness and looked at their effects on productivity. And so here, the blue arrows represent positive effects and the red arrows represent negative effects. And the size of the arrow is related to the magnitude of those effects. And so, because this is a little complicated, I'm going to break it down one by one. And so based on our first hypothesis that species richness was going to influence productivity through the fungal community, we saw that species richness did have a positive effect on productivity. So this supports the age-old idea that there's a positive diversity productivity relationship. Interestingly, the fungal community did indeed support this relationship by creating a positive indirect effect of species richness on productivity. But in contrast, the relationships with pathogens and ectomycorrhizae were both negative. So a negative times a positive arrow results in a negative indirect relationship for both ectomycorrhizae and pathogens. The role of functional identity in productivity was also very strong. And so the traits of the tree community had strong effects on the fungal community and the pathogen richness in particular. And these created indirect effects on productivity. And then water availability, surprisingly, had no direct effect on productivity, but it did have an effect indirectly through the fungal community. So water availability changed the fungal community composition, which influenced productivity. And this was a negative relationship, meaning that it dampened the effect of water availability on productivity. And then finally, if we look instead at complementarity, so this is a similar model, but looking at the magnitude of complementarity, so how much overproduction, in our case, we did see only overproduction, not underproduction, there was in these mixed species communities. We saw that species richness of trees was a very strong driver of complementarity. And again, that this was driven partially through the fungal community. So again, the fungal community supported the positive diversity productivity relationship. And here you can see that pathogen richness was not important in this model. And ectomycorrhizal richness actually had a negative, an indirect effect. And also interestingly, the water availability here, that there was no longer an indirect effect of water availability on complementarity. So water availability is not related to complementarity. So going back to the mechanisms of complementarity and those potential microbial drivers, we saw that there wasn't an effect of water availability on complementarity. So this makes us think that it's less likely that abiotic facilitation, at least in terms of water facilitation, is the main driver. And we also didn't see that pathogen species richness was an important component of that final model for complementarity. So that suggests that it may not be biotic feedbacks either, although there are ways that mycorrhizal fungi can promote positive biotic feedbacks. So instead, we suggest that it may be mostly resource partitioning that the fungal communities are mediating these relationships with diversity and productivity. But we can't rule out interactions between these three different mechanisms. So to summarize, we found that the soil fungi are promoting the positive diversity productivity relationship via complementarity, that soil fungi mediate the impact of water availability on productivity, but not the impact on complementarity. And because of the different components that we looked at, we suggest that this is probably due to differences in resource partitioning affected by the fungi. And so these have really important implications for a variety of different factors. So this can influence forest resilience under climate change, for example, as droughts become more common, and as we continue to lose biodiversity in forests. It also has important implications for forestry. So for example, if we can gain productivity and resilience to drought in planting more diverse forests, maybe this is something that we need to transition into away from planting monocultures. And most importantly, it demonstrates the essential role that soil biota and soil fungi in particular are play in the driving the diversity productivity relationship in forests. And so finally, I'd like to thank my collaborators on this project and our funding, and I'd be happy to take questions.
So yeah, hi, I'm Sarah. I just defended my master's thesis last fall at Purdue, and I'm still working with the Saunders Lab now before I move on to my PhD elsewhere. And yeah, so I also got the mycorrhizal memo, it seems. So I'm going to be talking about mycorrhizal fungal diversity in central hardwood forest stands that have been mostly managed for oak regeneration using prescribed fire. So just for a little background on the system and the project in general. So mature oak forests, like most ecosystems, are very complex, and there's many organisms working together. Oak ecosystems are ecologically and economically important, and they support many bird, insect, mammal, even spring ephemerals and amphibians, and as many different types of communities are best supported by oak forests. However, one historic piece of the oak forest ecosystem has been surface fire. which has mostly been kept off the landscape for about the past century or so, and can now be seen as more of a disrupted natural disturbance regime. So because of that, a lot of managers have been attempting to reintroduce fire to these systems that have not experienced surface fires for quite a few decades, and now mostly have pretty novel species compositions. So for the central hardwood region, that means mostly oak and hickory in the overstory, but then a lot of mesophytic species in the midstory, which hasn't really historically been recorded in this region. And another piece of the oak ecosystem is their symbiotic mutualism with mycorrhizal fungi. And oak specifically are ectomycorrhizal. So this project as a whole was kind of put in place to try to determine what happens to these fungi and their dispersal networks after the use of prescribed fire. And so fire science itself, and especially prescribed fire, it's pretty difficult considering it's so like site dependent. There's a lot of factors. And then that coupled with the fact that these dispersal networks are also pretty complicated. So it's been pretty difficult so far to parse out how mycorrhizal diversity is being affected by this style of forest management. And so this is also, I just want to highlight, this is just one kind of small part of a much larger project that's still ongoing to assess mycorrhizal community composition, diversity, and chipmunk-mediated dispersal networks and how all of that is being altered by the use of prescribed fire. So what I'm going to focus on in this presentation specifically is just what is present in the soil for the chipmunks to then consume and disperse. So because of that, we're focusing mostly on mycorrhizal fungi and also highlighting truffle fungi. So I'm using the term truffle pretty colloquially just to mean any fungal body that's going to fruit hypogeously, so below ground, since that is what in our preliminary examination of the chipmunk scat, that is mostly what they were consuming in this area. And you can see just this very basic schematic of this relationship. So image A depicts a mature oak forest, and then B, zoom in on their root tips, and then C, those colonized root tips will then produce fruiting bodies, truffles, which a small mammal will then consume, and then the spores will pass through their digestive tract and they'll get dispersed elsewhere. And then that will hopefully aid in seedlings regenerating outside of the mature root zone or the root zone of the mature forest. So this relationship is at least hypothesized currently to be, this dispersal network rather, to be pretty important. So to get into the methods, we extracted fungal DNA from 60 soil samples collected at the Hardwood Ecosystem Experiment, which is in South Central Indiana in the US. And then so with the DNA extracts, we used next-gen sequencing with the Illumina MiSeq platform. Then with all of our sequence data, we ran that through FunGuild and R to assign each of our taxa to a fungal functional guild. So that means it could be broken down. There's actually a lot of great information. I would recommend this to anyone interested in soil or just fungal functional guilds. But so I'm just going to focus on arbuscular mycorrhizal taxa and ectomycorrhizal taxa today. And then with the functional guilds, we then calculated measures of diversity using Hill numbers, which are basically the number of dominant taxa in the system. So to also frame how I'm going to talk about my results, as you can see on the left, that is just a snapshot of the diversity found in each sample. So as you can see, the samples are pretty diverse, and most of them contain many, many rare taxa. You know, if you really like squint and you can see all the little tiny little blips of color, those are each different taxa found in each of the samples. So to frame this talk, I'm just going to talk about 3 categories of variables that we measured: burn, abiotic, and biotic. And the starred variables are the ones that I'm going to talk about specifically today. Okay, so now to get into some of the results. So for 2 burn variables and how they affect arbuscular mycorrhizal fungal diversity. So first, burn intensity. So rare AMF taxa are preferring sites with a moderate burn intensity. And likewise, abundant AMF taxa are marginally preferring sites that were burned 3 years prior to sampling when compared to 4 years or unburnt sites. So basically the bottom line is that since both rare and abundant AMF taxa are preferring burned sites over unburned sites at the very least, we do have evidence that you know, they are kind of potentially contrasting their tree host burn preferences, considering in this region, the most common AM tree species like maple, black gum, and tulip poplar are all recorded to not respond so positively to the use of surface fire in this region. So at least right now, it appears that the mesification we see above ground may not be occurring below ground at our sites. However, we don't have any conclusive evidence yet for this, considering these forests here are AM dominated. Really only oak, hickory, and beech are the big more dominant EM species here, and basically almost everything else is AM. So it could just be another AM tree species besides the very common mesophytic ones that are driving this pattern. Now moving on to our abiotic factor of litter layer depth and how that is affecting our truffle taxa diversity. So all three orders of diversity, so rare, so for the Hill diversity numbers, so Q0, 1, and 2, so rare, average, and abundant truffle taxa are all affected by a change in litter layer depth. But it's interesting because rare and abundant truffle taxa are actually opposing different levels of litter. So as you see, rare taxa are preferring a moderate to high level of litter, but abundant truffle taxa are preferring a lower level of litter. I mean, that's especially interesting because the literature suggests, and if you've ever just gone out and looked for truffles yourself, you'll know that they can often be found within the litter layer. So usually they're associated with a higher litter layers, giving them, more habitat, more access to nutrients. So the fact that abundant taxa are preferring a lower litter layer is pretty interesting. But also on these sites specifically, a lower litter layer is often found on the stands that have been burned. So that also might be an interaction occurring that giving us this pattern as well. Okay, and then moving on to the last variable, so how the shade tolerance of locally dominant woody plant species is affecting arbuscular mycorrhizal fungal diversity. So we found that the AMF community as a whole, or the average AMF taxa, prefers shade intolerant dominated plots. And again, that's pretty interesting considering two of the most prevalent shade intolerant trees at these sites are oak and hickory, and those are both EM tree species, not AM. So again, that kind of brings me back to mesification, since it has been recorded over the past few decades that these sites are at least above ground, very entrenched in a mesification cycle. So it's interesting that we're seeing this kind of odd pattern below ground, considering we can't really pinpoint what shade intolerant tree species might be driving this pattern, considering, like I mentioned, the shade intolerant oak and hickory are both EM, and most common mesophytic species at our sites are AM, but they're shade tolerant. So it's just kind of difficult to explain this pattern by just looking at the locally dominant plant species alone. So again, it's probably some sort of interaction or maybe another variable that we just haven't measured yet. Okay. So then to pose some more questions and kind of give an idea of where we're going next with this project. So is mesification really not occurring at our sites below ground, or have we just not burned enough to break the cycle? So all of our burnt sites had only had one fire at the time of sampling. And the above ground literature is kind of wishy-washy, I would say, on if one burn is enough to break a cycle. I think it's just very, like site dependent on what is actually going on at your site, if one burn would be enough. So I think that there needs to be at least 2 to 3 more burns. And then if we reassess the soil fungal community, we might get a better idea of what is actually happening after burns. And also, like I've been mentioning, the measured burn variables are definitely, they're influencing and are also influenced by the biotic and abiotic variables. So once we do have all the data, we're going to build much more complex models to better incorporate these interactions between the variables. And also, so we have soil chemical data that we can also incorporate into those models. And we have chipmunk fecal pellets that are being sequenced, hopefully as we speak. So then we can then incorporate that information. So we'll have what is actually present in the soil, then we'll have what is present in the chipmunk scat to then compare if they're preferentially consuming certain taxa over another before or after a burn. And that is all I had, and I'd like to thank all my funders listed down there, and I will be happy to take any questions.
I'm presenting on work we published earlier this year, which looks at ash application at multiple AshNet sites across Canada in a meta study. We had many authors on this site. There's a lot of sites across AshNet, so we had a lot of contributions from anyone involved with any of the sites used in the study. Before I discuss AshNet, I just wanted to talk about why we use ash in forests and why we might want to use ash in forests in the first place. We've had a lot of talks in this conference about emulating natural disturbance. Ash application could be used to help emulate fire disturbance in harvested sites. Ash itself has a lot of components that make it a possibly good amendment. It's high in calcium and magnesium. Some ashes are high in carbon. And it's alkaline, so it can raise soil pH if you have acidic soils. Also, ash is currently being landfilled. It's classified as a waste product. So if we can find an alternative use for it, we can save some landfill costs from the bioenergy sector. AshNet's a series of experiments which assess whether ash addition to forest systems can improve forest health. While there are differences in how these individual studies have been executed, they all apply ash to manage forest. You can see on the side here a picture of one of the sites with ash being hand applied. These studies have differences in ash type, stand composition, plot sizes, parent materials, but despite all of this, we can still use them to assess if there's broad changes to soils across eight different sites. Across these studies, we had a variety of ash qualities. Ash chemistry is different due to different boilers and feedstocks at each site. Some of the differences are due to the type of ash. For example, bottom ash is in higher carbon and typically has larger particle sizes than fly ash. In this graph, the ash added sodium to each site in kilograms per hectare is on the y-axis, and the ash added calcium in kilograms per hectare is on the x-axis. Each point is a unique site treatment combination. And you can see that we have a range of ash qualities and ash amendment rates. We can use these to look at if the components in ash, whether they're possibly beneficial or antagonistic, are driving generalized effects across these sites. It's not surprising that we have a lot of site differences since these sites are spread across the country across spatial gradients and in 3 main forest types. But since they're all part of the AshNet network, they have the common study design of having control and ash paired treatments and paired plots. With these, this allowed us to piggyback on the existing study designs and collect new soil samples. With these new samples, we performed a kind of meta-analysis, but with real samples that had consistent sample processing and statistical methodology. In short, we sampled soils from all the treatments on all the blocks in each of these sites, and we assessed these, the different horizons in the soil for enzyme assays, for activity of nitrogen and phosphorus utilization, and we extracted DNA from them and performed metabarcoding for 4 different target genes, 16S for bacteria, 18S and ITS for fungi, and F230, which is the CO1 gene for arthropods. These samples were also analyzed for soil quality indicators in another companion study that's going to be talked about later on in this session. And they gave us the total C and total N to use as explanatory variables to see if there was changes in community compositions or in nutrient acquisition. First, we wanted to see if we could detect any broad effects of ash. Because we didn't have replication within block, we had to ignore spatial variation because we couldn't include it as a random effect in models. But we still could run models across the sites to look at if ash calcium or ash sodium applied being explanatory variables in ANOVAs, RDA, and GLMs could explain any of the patterns we saw in the dataset. We're running tests for the enzymes, diversity metrics, composition of communities, as well as for individual organisms. And these tests are repeated across each of those metabarcoding targets, and we did them at several levels of grouping. So we assessed at functional, genus levels, and ASV, which is unique sequence variants. This ends up being a lot of statistical tests, which does make it our analysis kind of biased to having false positives. But I wasn't too concerned about this, as you'll see from my results. So the reason why is because we generally are not finding any significant results. So false positives is less of a concern than false negatives in this analysis. For phosphatase and nitrogen utilization, our ANOVAs showed that ash and the ash amount did not have significant effects for either phosphatase or nitrogen utilization. That's after accounting for site and soil type. And when we looked at diversity metrics, this graph summarizes the results of all of the diversity metric ANOVA tests. There's the probability of the effect of each parameter included in the ANOVA along the bottom, and the parameter that was included in the ANOVA is along the side. There's different colored bars representing each diversity metric we assessed, and there's a different panel for each metabarcode that we assessed. There's also a black line on each panel that represents P equals 0.05. And you can see that none of the diversity metrics had a significance lower than P equals 0.05. So we also didn't have any significant trends in diversity metrics in response to ash. We also looked at community composition. This graph shows the proportion of variance explained that can be explained by ash quality parameters after accounting for site and soil type using a partial RDA. This type of analysis was conducted for each of these metabarcode grouping levels. And for all of these data sets, you can see that we don't ever explain more than 2.5% of variance in the data set by the ash parameters. When we look at how that 2.5%, well, less in this case, that 1% of variance is distributed, we can see that those really low explanatory axes are really driven by only a couple, two sites driving all the difference in that small proportion of variance. So we're not seeing broad trends on community structure. Here. And then we also weren't detecting any specific changes in organisms using ALDEX analyses. So any of these small effects here that are specific to certain sites are spread across multiple organisms within those sites and not specific to, not driven by one or two organisms having larger changes. Because there's a chance that we're not detecting effects because we didn't account for within site variation, which we know can be very large. We also ran pairwise tests within blocks to compare. Sorry, we ran pairwise tests by calculating the difference between controls and blocks and treatments within blocks, and assessed one-way t-tests or Wilcoxon tests of the difference between those parameters. We did this for all enzymes and diversity metrics, and then we also did pairwise t-tests using paired controls and blocks. This ends up being a lot of individual tests, so again, we're still very biased to find false positives. This graph shows the difference in N-acetyl glucosaminidase activity, which was used to assess nitrogen utilization. The box plots show the distribution of the calculated difference in activity between controls and treatments within a block. And they are colored solid green if they were highly significant. So that's a Bonferroni corrected significance of 0.05. An empty green box plot if they were significant at 0.05, and just gray if they weren't significant at all. You'll see that there's some significant results, but they're in different directions and happening in different layers of the soil. And the direction of the effects is also different. There are even some different responses at Eliza Lake, which were the most similar studies and used the same ash. So there was no consistent effects across sites. This graph that shows phosphorus utilization can be read as the same as the previous. And there were even fewer effects here. So no consistent effects across sites for phosphorus utilization either. This graph summarizes the result of every paired community t-test to see if there was any significant group within any of the within any of the metabarcoding data sets for any particular grouping level. And there was not even one for any particular site that was significant, that had a significant response to ash. So there was also no effect for any individual organisms or taxa. So basically, we really didn't see any ash-based effects that were large enough to detect a general response without digging into the data set. So we're looking at really small amounts of variance here, and the groups that are being impacted aren't impacted in extreme ways that we can see, i.e. losses of or introductions of novel organisms or groups. But we did see some changes in enzyme activities, but these were different and very different between sites and site specific. So there seems to be an interaction between site and ash quality, an ash type that might be causing those responses. And that's everything for my presentation, so thank you.
So thanks for being here today virtually. So I'm going to talk about early forest composition response to soil treatments in a sugar maple dominated stands. And first of all, I want to acknowledge my co-authors. There's David Paré and Fidel Bougnouniou. Fidel is the one who did most of the analysis here. So sugar maple, the species is found in temperate deciduous forests of eastern North America, and as you may know, has a significant importance. It provides many ecosystem services, including quality wood for flooring, furnitures. Its sap has been used for centuries for maple syrup production. Unfortunately, a decline in the vigor, growth, and abundance of sugar maple has been documented at multiple sites in recent decades. This decline has been associated with various drivers, including soil acidification and consequent losses of base cations, calcium depletion following harvesting, severe meteorological events such as droughts, freeze-thaw cycles, defoliation events, as well as competition with other tree species. And one competitive species able to replace sugar maple stands and lead to regeneration failures is American beech. Indeed, the species perform quite well on acidic and poor soils and has a high shade tolerance and the ability to reproduce vegetatively by root sprouts. And one problem with beech is the beech bark disease caused by a fungal pathogen. So the disease causes severe cankering, as you can see in the picture over here, deformation of the stems as well, and eventually tree mortality. So the disease reduces or even prevent wood use in products, and can also decreases the amount of forage trees for wildlife. So what are the potential solutions to reduce or control the dominance, the competitiveness of beech over maple and other hardwood species? So we can remove beech. We can increase light availability by creating gaps, partial cuts, so that species with higher light requirements can take advantage. We can increase soil pH and improve soil fertility with inputs. Because, as said earlier, beech perform quite well under acidic and nutrient-poor environments. And finally, we can disturb the soil surface to facilitate seed germination and seedling survival of some species, but not too much because severing beech roots can trigger sprouting. So there's a need to develop management strategy to control American beech expansion, promote the regeneration of high-value species, and also help reduce the impact of beech bog disease on stand development. So the goal of this research here is to assess the effect of soil treatments on the seedling recruitment in a sugar maple dominated stand prone to regeneration failure. And the question we ask are first, what factor drive seedling recruitment? And second, is there a treatment alone or a combination of treatments that can control beech regeneration and promote other species at the same time? So the study is located in Eastern Canada at Valcartier near Quebec City. And the site is part of the AshNet network of ash application trials. We established the trial in a mature 70 years old sugar maple dominated stand with yellow birch, American beech, and red maple, as well as pin cherry as companion species. The stand was affected by beech bog disease and showed evident signs of maple regeneration failure. That is a significant portion of the ground cover without regeneration, very low abundance of maple seedling, and a high density of beech saplings. The soils are classified as district Bruni soils developed on sandy loam till. And in 2014, the stand was subjected to a selection cut, which removed 25% of the initial forest cover, including most of the beech trees. So we established 12 plots divided into 8 4-square-meter subplots in which we applied our different treatment for a total of 96 subplots. We have 4 soil amendment treatments: control, lime, and wood ash apply at a rate of 5 tons per hectare of calcium carbonate equivalent. We have fertilizer addition as ammonium nitrate. We also have 1 mechanical treatment consisting in a light manual scarification, and the result can be seen here on the picture on the lower right. And this treatment is applied or not in combination with the amendment treatments. So as said earlier, we are conducting this trial in following selection cut because maple and birch tend to be favored by canopy openings relative to beech, and we want to see if these additional treatments can effectively favor these species over beech. We also remove all beech seedlings and saplings from the plot prior to treatments. And what do we measure? Seedlings abundance within 4 different size classes. Inventories are conducted 2 times a year. We also monitor multiple environmental parameters, including light availability, soil moisture, texture, nutrient availability, and companion species. So now to look at the evolution of regeneration with a visual support for a single subplot. The first picture in 2017 over here in the upper left is before treatment, and we can see a beech seedling in there, which is here. So following the treatment, the year after, in 2018, the beech was removed, like I said, and this subplot received a lime treatment. So it might be difficult for you to see, but there are little maple seedlings appearing in 2018 where the beech was growing on the lower left corner, and they are still there in 2021. Unfortunately, the beech came back on the upper left corner of the quadrat in the 2020, and it's been expanding since then. So now the result of a multivariate analysis, which reveals high dissimilarity among species recruitment based on environmental variables that include soil and residual vegetation characteristics. We studied the regeneration of multiple tree species, 7 of them actually, but today, for the sake of time, we will focus only on 3 of them. So yellow birch here, beech, and sugar maple. So our analysis shows that yellow birch and beech regeneration are negatively related with each other, while sugar maple regen is poorly related with that of beech or birch. And maple regeneration is more related to soil moisture and texture, as seen here. So now if we look at the main drivers determining species recruitment with a random forest analysis, we show that those drivers are species specific and can be of biotic, abiotic, or experimental origin. And I label the drivers with different colors in the upper right here. So abiotic is blue, green for biotic, and red for experimental, so the treatments. So there's a lot of variables in here, and we're not going to look at them individually. So I'll make a summary for you. So for yellow birch here, the soil treatment and especially scarification is very important, along with the presence of pin cherry, light availability, and soil texture, whereas amendment is not a major factor. For beech, now light availability is the most important, also soil moisture, the presence or rather the absence of birch, cherry, and balsam fir, and the factor treatment, which is here the interaction between amendment and scarification seems quite important, whereas the amendment and scarification are less important. Now for sugar maple, the main factors are moisture, light, texture, along with the interaction of amendment and scarification. So overall, the soil treatment had the high influence on yellow birch regeneration and a moderate influence on American beech and sugar maple regeneration. Now focusing on the species response to soil treatments, we found that the strength and direction of the response is species and time dependent. In the graph, we have the change in the seedling abundance over time from 2018 to 2021, and for 4 size classes, so 0, 15 centimeters, 15 to 60, 60 to 130, and above 130 centimeters. And the different colors, they represent the different amendment treatments. So green, it's control, orange for fertilizer addition, lime for blue, and wood ash for gray for wood ash. And the hatched bars are for scarification. So in summary, we see for yellow birch that scarification really dramatically increased birch seedlings, while amendment, either lime, fertilizer, or ash, had a negative effect. Now for beech, on non-scarified soils, fertilizer and lime application reduced significantly beech recruitment, while ash amendment had the opposite effect. And for sugar maple, lime and wood ash, the blue and the gray bars promoted seedling abundance relative to control. So in conclusion, our results suggest that a combination of soil scarification and amendment could help fine-tune early forest composition and dominance in these forests. Unfortunately, none of the treatments investigated here was able to control beech and promote the regeneration of other hardwood species at the same time, so no one-size-fits-all treatment. Biotic and abiotic variables certainly play a role and interact with experimental treatments to drive species regeneration. And it remains to be seen if the current species composition will be maintained over time, if other treatment-induced impact will appear later. So we'll monitor that for the next few years or decades. So thank you for your attention, and special thanks to all the persons listed here who were involved in this project.
Well, good afternoon. Great talks by everybody this afternoon. It's really exciting to be back in the forest and to hear about the diversity of things that are happening across North America. So I'm really excited to be here to share the work of a former master's student in my group, Ruth Joseph, that was recently published in Global Change Biology, Bioenergy. And I want to thank our co-authors, particularly Lisa Vanier and Dave Morris, for their patience and guidance, and Paul Hazlitt for spearheading AshNet, and of course the Program of Energy Research and Development for ultimately funding this work. So given the climate emergency, there is some urgency to pursue alternative and renewable sources of energy. And not overly surprising is that the pulp and paper industry has been generating energy and electricity through the combustion of the residuals, bark, and wood waste for decades. So what's left over, of course, is ash, which sadly is typically landfilled. And it's landfilled for a whole host of reasons, but a key one is regulations. So when we burn biomass, it concentrates metals, which of course is a concern for environmental quality if we're going to be spreading it on the landscape. And as an example, forested systems in Ontario, land application is permitted through the Ministry of Environment and requires an environmental compliance approval. And each province has different requirements. So it can be a really frustrating sandbox to play in because fire, as we know, is a natural process that generates ash, and returning ash to a site that, let's say, was harvested could be a way to replace nutrients removed from the site through harvesting. And ash, as Emily mentioned, is an alkaline material, and increasing pH combined with that return of limiting nutrients like phosphorus and potassium may improve site productivity while increasing economic productivity as well. But of course, what I'm really interested in is the potential to increase organic matter stores. So anyone who knows me knows that I'm pretty passionate about soil organic matter. Soil organic matter is the largest terrestrial reservoir of organic carbon, but it also provides many, many ecosystem services. And it's really what I view as the heart of the soil. So soil quality is a concept that's getting a lot of attention lately. It's defined as the capacity of a soil to function for specific land uses or within boundaries. And again, this has been getting a lot of attention because of concerns around soil degradation. So soil quality, it promotes environmental quality, which will in turn hopefully contribute to creating sustainable systems, both economically and environmentally. So this whole concept of soil quality, some have argued that it's
It's just a metaphor, but in reality, the attributes of soil quality or indicators of soil quality that we use can be really helpful to reveal how land management practices are impacting soil functions both in time and space. So soil organic matter is a key attribute in most definitions and measurements of soil quality because it plays such a, well, it just has such a huge influence on the physical, chemical and biological properties of the soil. So the logical question for me is, does the land application of wood ash to soil affect soil quality? So in Canada, the last couple of presentations have talked about sites in the AshNet system. So we have this dream team of scientists, foresters, industry, and government reps that we share this common passion for reducing waste and improving forest health through the application of wood ash from bioenergy to forest soils. And Lisa Vanier was leading the metagenomics project that Emily talked about, and was kind enough to collaborate on a study looking at the responsiveness of indicators of soil quality to ash application at 8 sites in the AshNet network that include and span 5 provinces in Canada. So soil samples were collected in the spring, summer of 2017, and collaborators collected samples from sites that were receiving wood ash, and you saw some of that design earlier. So the forest floor was removed using a 15 by 15 centimeter square, and we can see that indicated in the picture delineated by those 4 golf tees. And 3 PVC pipes were used to extract the top 20 centimeters of mineral soil. So the forest soil was separated into layers as appropriate. And all samples were frozen and shipped to the Great Lakes Forestry Center in Sault Ste. Marie. And they in turn sent us a subsample of those samples to work our magic. So I'm assuming that most of the audience works in forest soils and appreciates the variability and all of the joy that comes with it. And detecting management-induced changes is really tough because this pool is big. There's a lot of spatial variability, and trying to detect these small changes can be really challenging. But these indicators that are known to be sensitive and represent important functions can maybe shed some light on what is happening. So we measured 12 different indicators. Some were calculations, and they're all meant to really represent soil function. So carbon to nitrogen, something we're all familiar with, reflects the capacity of the soil to store and recycle nutrients. Hot water extractable carbon is basically a tea that we make. It's really rich in carbohydrates and is known to affect soil structure. Microbial biomass is just this, gross estimate of the microbial community, but it is quite responsive. So the idea is that if the application to wood ash to soil is influencing soil quality, then it should show up in these indicators. So easy peasy, right? Well, not so much. And the aspect that I think that we, and I'll just put it all on me, that I struggled with most was the difference in chemistry across the ashes, which I knew about, but the similarity in the range of the rates of application. And to really kind of make this more clear, we see similarity, and I highlight that in the slide, in the rates of wood ash applied across these sites and in the literature, but what do these rates of application, really represent from an elemental or a stoichiometric perspective? So some ashes are pretty well characterized, so we could maybe convert some of these rates of application to evaluate things on an elemental basis, but others are really less so. So I will give a little whoop whoop special shout out to the Island Lake group because they applied wood ash at rates to represent a reapplication of calcium to reflect what was removed in a harvest, or kind of on a scale of that. So for Ruth's thesis, she just treated each site individually, meaning that she ran the stats for each site and each soil layer to test for differences. So out of the 8 sites, multiple soil layers, 12 indicators, which accounts for several hundred tests, there were only 16 significant differences. And Emily talked about the Bonferroni correction earlier. And if we take that into consideration, or the potential for that type 1 error, there were actually only 3 significant differences in this data set. So of course, we wanted to publish this work. It's a really nice comprehensive data set. And Lisa and I had discussions about looking at effect sizes. And that's something that I had thought about when in guiding Ruth. And in fact, this is something that came back from one of the reviewers. But again, there's not really that replication. And Emily talked about that. So I puzzled and I puzzled and I puzzled some more. And then Dave thought of something that I hadn't before, and we settled on a staged approach to the data analysis. So with the staged approach, we looked to ask questions where there was some level of replication. And this involves some grouping of forest floor layers and rates of application to ask, basically, does ash type matter? So we looked at the group of BC sites. Does soil texture affect the response to ash addition? And thirdly, is there an effect of ash application on mature hardwood stands? So I could blind you with science, but honestly, I think the title of the talk was a bit of a spoiler, and I'm conscious of the fact that your brains are probably full up right now, given that I am the last talk of the day. So the main upshot of this story is that there were really very few effects, whether we took the site-by-site approach or the staged approach. And I'm not gonna lie, it was frustrating. It was frustrating in the sense that there were really no, there's no money picture here. There's no money graph. There's no earth-shattering effects. So we can really go through and analyze this data out the wazoo. But at the end of the day, the story is more or less the same. And in many cases, the differences we reported were significant only when we relaxed the p-value. There was a lot of interaction, and really the differences were site-specific. So taking a step back to really look at that big picture, because, no results aren't fun. There were no consistent effects of wood ash application on measured soil organic matter attributes across the sites, but labile indicators did seem to be more responsive. When significant differences were observed, they were often close to the surface. I use close to the surface because not all sites had a really well-defined or thick organic or forest floor layer. And overwhelmingly in the staged approach, the greatest effects were actually between sites. And by that, I mean the attributes didn't differ with respect to treatment. They really just differed between sites. And then, that opens up a whole different can of worms, because that wasn't the purpose of the study. We know these sites differ. So a big learning lesson for me through this work was that was to really give pause for thought when evaluating the effects of land application of these types of amendments. And each wood ash or pulp mill residual is different. But when we look in the literature, we often see these really nice round numbers for rates of application, which makes me think I can compare, but really, can I? And it actually makes so much more sense to start thinking about testing for rates of application that actually make sense from a nutrient return perspective, which is what will actually determine the rate of application in the real world. And despite my passion for organic matter, I do recognize that there's other parameters like heavy metals. But the rates of application that we actually do make on the landscape are limited by the chemistry of the ash and the soil itself. And we can't apply at levels that would be considered toxic. And in actuality, the rate of application of these materials is limited by the nutrient content of the materials themselves and not the heavy metal content. So I think the biggest contribution of this study is demonstrating that there really are limited effects of land application. But when we did see effects, they were generally positive. And I would argue that if it makes economic and logistical sense to return this material to the land and perhaps even apply it more often. So if you're interested in looking at the differences, I refer you to the open access publication in the spirit of shameless self-promotion. And thank you for listening.